Well, it's great to be here. Really appreciate the opportunity. So as Chris said, I'm a program manager at Department of Energy's Advanced Manufacturing Office. And for the past 4 years or so, I've been building out a program in projects on atomically precise manufacturing. We have a portfolio of, I've lost count of the number of projects, but it's $18 million of funding for atomically precise manufacturing. So I've been really happy about our ability to do that. This morning, I'm just going to give you a very brief overview of atomic precision, just so we're all on the same page when we use this term. What does that mean? And why is it important? Why are we motivated to use this approach for longevity? I'll be giving some examples from some of the projects that we're funding and hopefully build a case for using molecular machines. So what is atomic precision? What it really means is getting every atom exactly where you want it to be. No defects, nothing out of place, virtually perfect materials and devices. For now, when we talk about atomic precision, we're really talking about molecules and assemblies of molecules, or maybe atom by atom positional assembly, putting every atom where you want it to be. And there's no size restriction on those structures and devices, except for what's really practical to be made. And I put up some complementary terms here because particularly in the area of nanomedicine and longevity, I'm personally confused by the terminology I've been seeing. So I'm going to try to deconstruct some of this. This definition for nanotechnology, the design, characterization, and manufacturing of structures, devices, and systems, and manipulating at the nanometer scale, there's a number of similar definitions out there for nanotechnology. This one was taken from a conference brochure on precision nanomedicine. What they really mean is nanoparticles, pretty much, when they talk about nanotechnology in the general community. And I sit on these interagency bi-monthly meetings with all the other feds, with the National Nanotechnology Coordination Office, in support of the National Nanotechnology Initiative, and by and large, that's it, nanoparticles. And then you've got nanomedicine, the application of nanotechnology in a healthcare setting to prevent and treat human disease. Okay, with nanoparticles, I guess, right? And then I got really excited. This term popped up just a few years ago. Precision nanomedicine, right? I thought finally they're getting right. They've drunk the atomically precise Kool-Aid. Like, no, they didn't. What this is an emerging approach for disease treatment, accounting for variability in genes, environment, and lifestyle, and again, nothing about atomic precision. Now, drugs, pharmaceuticals, they're molecules, right? And they're atomically precise, but clearly that's not what we're talking about at this workshop. We need to be talking about something different than what everyone's already doing. And in that regard, I've put up this statement. We're talking about dramatically improved capabilities to make, manipulate, and react molecules. And I'll get into that a little bit. So why are we so concerned about using atomic precision in the context of improving longevity? And I've just listed a few reasons here, and I'm sure there's a lot more that we're going to talk about over the weekend in this workshop. One is to repair cell structures that are important for longevity. We've just heard a great introduction to the effect of accrued damage and things no longer working the way they should be working. And if you can repair these molecular structures to atomic precision at the molecular level, that's going to result in unprecedented advances in health and in longevity. We also might want to remove undesirable structures that accumulate without damaging the healthy cells. I've put up this example of beta amyloid plaques that are related to the degeneration of the human brain in Alzheimer's patients. Wouldn't it be great if we could just get rid of those and not harm all the other cellular structures around them without even understanding how they function? And something that came out of our last workshop on longevity, the ability to use atomic precision as filters to remove toxins, to remove undesirable chemicals from the body that are helping to cause this degeneration that we're trying to prevent. And finally, we want to use atomic precision to develop new tools that will provide advanced capabilities for doing all of this stuff. Oh, one thing I wanted to mention here. There's a problem out there with the media, because you'll see a lot of images and articles about nanorobots repairing cells and structures in the human body. This is just one of many that I came across in preparing this talk. And the way it's presented in the media, it's as if these nanorobots were already here and already repairing stuff in the human body, and they're not, right? So it came as quite a blow. I gave a talk at it's not Comiccon, but it was like Comiccon in Washington, D.C. a couple of years ago, and these poor folks had expected that we would have nanorobots to show them that we're out there fixing the human body, and it's like, no, there aren't any. We don't have them, and I was sad to let them down a bit in their expectations, but this for us, this is a problem, right? Because people are already expecting that technology is here that isn't here yet. We're trying to find ways to fund it, and if people think we already have it, it's even harder. Okay, so just kind of an atomic precision 101, right? How do we make things that are atomically precise? Well, we make molecules, right? We're pretty good at that, and they are atomically precise and are all the same, and we're able to do that in great quantity and high yield. And there are some methods that have been emerging that allow us to make molecules that can serve as building blocks for larger structures, and that means they have to be conformal, be able to snap together. And so there are techniques like molecular Lego, Chris Schaffmeister at Temple, there's DNA origami, there's protein engineering. So there are ways to make molecules, not just molecules, but molecules that can be used as building blocks to make larger atomically precise structures. So what are the ways to do that? Well, one is self-assembly, right? You want to be able to design a molecular building block that can snap onto copies of itself without any defects. So it completely fills in, tiles very well, and then you have a product that is atomically precise because it's made of atomically precise building blocks, but it's much larger than the individual molecules. And I've shown an example here from our friends at Covalent, who are represented by Martin Edelstein and Gail Pergamut, who have developed a technique to do that, to design triangular molecules that fit together and be able to form a membrane that is one molecule thick, and all the pores in the membrane, because each pore is a molecule, all the pores are the same. So you have 100% selectivity. You've got a permeance that's through the roof because there's almost no resistance to flow. And these things are covalently bound, so they're strong and thin and atomically flat, and almost nothing sticks to them. So a beautiful example of how to make atomically precise stuff. The other way is positional assembly. So you don't have to design molecules that snap together as elegantly as in an atomically precise membrane, but they do have to snap together somehow. And how do you get those molecules, or even if you're doing atom by atom positional assembly, individual atoms, how do you get those to come together? And there's two ways that people are going about that. One way is using scanning probe technology. And the other way to do individual atomic either abstraction or assembly. And the other way is you have a molecular machine that can grab a hold of a molecule, bring it to a reaction site, react it with another molecule, and then molecule by molecule build up atomically precise structures. Okay, so I talked about the one example here on water purification, atomically precise membrane for water purification. Chris Schaffmeister at Temple is also working on atomically precise membranes, same approach to do separation of hydrocarbons. Simple, right? Except that it's not so simple. It's a real hard design problem. And then there's advanced self-assembly, right? So nature, biological systems, exploit this technique, self-assembly, to make some pretty impressive stuff, right? On the left here, I have an example of a bacterial flagellar motor. It's kind of an exploded view, so you can see how the individual proteins are assembled. They have complementary shapes and charge distributions to allow them to snap together in just the right way, and that's how they come together to make a motor for bacteria, flagella, that can go and swim about. And also this thing that looks like it came out of a spare parts factory for robots is it's a virus. It's a T4 phage, and it also is made by self-assembly of the individual parts, the tail fibers and the head and the the collar and the end plate and so forth, there's a sequence by which they do self-assemble in solution to create essentially a robot that infects bacteria. But these are really hard, right? Because these proteins that snap together, while they work really well, this is the product of a billion years of evolution. So it's taken some time to get to that level of elegance. And it's hard for us to do protein design to make atomically precise stuff by self-assembly. I mentioned the use of positional assembly with scanning probe microscopes. We have some projects that we're funding at Department of Energy. The first one is with Zyvex Labs, and they use a technique that our co-chair, Joe Lighting, has pioneered to abstract hydrogen atoms from the surface of a crystal and then bring in a gas and essentially put atoms into those reactive sites. We're also funding work, and their target application is quantum electronic devices. We're also funding work at University of Texas, Reza Mohammadi, who wants to parallelize this technique, to have literally thousands of these scanning probe tips doing atom by atom assembly. Folks at UCLA, Jim Kinjewski, has a slightly different approach where instead of doing hydrogen abstraction of a surface, in this technique, we're talking about taking a molecule and moving it to a reactive site and causing a reaction to occur. That's been done, right? In 1999, Ho and Lee at Cornell used a scanning probe tip to pick up an individual carbon monoxide molecule. They moved it to an iron atom sitting on a surface, passed a current, and created a chemical bond between the iron atom and the carbon monoxide molecule. 1999. Recently, you'll see a similar piece of work on a molecule that was theoretical but couldn't be synthesized using conventional chemical techniques, but they were able to make this molecule called triangulene by positioning molecules and causing reactions at particular sites on a surface in a vacuum. Okay. So, synthesis, atomically precise manufacturing using molecular machines. We're funding a project in this area, Dana-Farber Cancer Institute, William Shea, in conjunction with Andrew Turberfield at Oxford, have proposed a couple of different techniques to do this. Essentially what they want to do is use DNA origami to make 2D XY printers, atomically precise nanomachines that can pick up molecules and react them at specific sites on a canvas. And they want to spell out molecular additive manufacturing, MAM, as one of their project goals. It seems kind of strange. We're talking about DNA, right, which is a helical molecule, but these highly regular structures using folded DNA. But that's been done, right? So here's the largest structure that, at least as of a few years ago, that was made of 51,000 base pairs of atoms in these interesting rectangular shapes. now we're to the point in the talk where I get to complain about Microsoft. Because I had this working, honestly, in my hotel room just before I walked over, working embedded YouTube presentation. And it no longer works. I don't know. Maybe somebody can help me figure it out eventually. But I have a plan B, which is to just go right to the web browser and YouTube. So I'm going to, this is a molecular machine, it's a ribosome, that makes proteins by reading essentially a numerical tape and doing additive manufacturing, positional assembly. And so I'm just gonna run this. It only takes a minute.
When the RNA copy is complete, it snakes out into the outer part of the cell. Then, in a dazzling display of choreography, all the components of a molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein. Special transfer molecules, the green triangles, bring each amino acid to the ribosome. The amino acids are the small red tips attached to the transfer molecules. There are different transfer molecules for each of the 20 amino acids. Each transfer molecule carries a 3-letter code that is matched with the RNA in the machine. Now we come to the heart of the process. Inside the ribosome, the RNA is pulled through like a tape. The code for each amino acid is read off, 3 letters at a time, and matched to 3 corresponding letters on the transfer molecules. When the right transfer molecule plugs in, the amino acid it carries is added to the growing protein chain. Again, you are watching this in real time. And after a few seconds, the assembled protein starts to emerge from the ribosome. Ribosomes can make any kind of protein. It just depends what genetic message you feed in on the RNA. In this case, the end product is hemoglobin. The cells in our bone marrow churn out 100 trillion molecules of it per second. And as a result, our muscles, brain, and all the vital organs in our body receive the oxygen they need.
Hemoglobin. All right. Yes. Isn't that amazing? That's the power of massive parallelism, right? So there's about 30 trillion cells in a human body, up to 10 million ribosomes in a human cell. So there's enough to do it. That was pretty cool, wasn't it? Real time making a hemoglobin. Okay, so that's advanced positional assembly with a molecular machine. So speaking of longevity, there's no reason in principle under the right conditions that molecule can't last a long, long time. I just have a couple of examples here. This, in this case, researchers found bacteria that were held in permafrost in Canada that were about 500,000 years old, 500,000 years old, intact bacteria. In this case, this is very recent, pigment molecules were found in fossils intact that were 350 million years old. So molecules can last a long time, but the problem is that in living systems, especially living systems the size of us, you need repair mechanisms because those molecules don't last in that environment. thus the case for molecular machines. So you need a technology that's going to be able to bring cellular structures, molecular structures, back to their pristine state and reverse aging. And I think molecular machines are a key enabler of that. I've got one more painful switch over to, or no, that was it, to just illustrate a couple of repair mechanisms, molecular machine repair mechanisms that are already
UV
in
light
place.
and other ionizing irradiations damage DNA molecules by creating thymine dimers between adjacent thymines in the DNA chain. The dimers weaken hydrogen bonding and lead to deformations of the double helix that create subsequent errors in replication, resulting in mutations. To protect cells against such damage, DNA photolyase, a light-dependent DNA repair enzyme, eliminates thymine dimers from the DNA strand. The enzyme binds to a thymine dimer, and upon photoactivation, transfers an electron to the dimer, leading to the reformation of two thymines. The electron is subsequently returned to the enzyme after the dimer has been eliminated. Chemical agents can induce the methylation or alkylation of guanine residues in DNA at the oxygen attached to the 6th position of the purine ring. These methylation changes can result in mutations. The enzyme methylguanine DNA methyltransferase eliminates these methylation events and restores the correct structure to guanine in DNA, preventing mutations from occurring.
So a bit dry presentation, but I think you get the point, which is that molecular machines are capable of doing extraordinary repair. But clearly it's not enough, right? Because we still get old and we lose our health. So there's much more to be done. And I think the answer is in advanced molecular machines. Here's a concept from an animation funded by NanoRex. You can find it on YouTube, in which advanced molecular machines can grab onto molecules and use them as building blocks for other atomically precise structures. I just have a quick closing thought about this. And we're going to be talking about a lot of really cool stuff over the rest of this weekend. And it's easy to get all caught up in that. But I would say that while pretty much everybody here, I'm sure, is very enthusiastic about the prospects for improving longevity, for essentially reversing aging, that doesn't necessarily play well in the general public. You know, there's always folks who are going to say, oh, that's science fiction, or oh, you're playing God, right? Oh, if we stop aging, then the planet will fill up with people, and that's unsustainable. And you know, the folks here, Chris and Gail, and a lot of people in this room have come up with, I think, pretty good defenses to those sorts of criticisms. But I do want to just kind of remind everybody that that's out there. That while we're enthusiastic about moving this technology ahead, me included, you know, I'm kind of a big chicken about it, right? I'm at the Department of Energy. We're funding programs on atomic precision. I don't talk at all about longevity, about health, because it's safe for me to just say, I'm just going to stick to our mission statement, energy efficiency. I can completely justify a program on atomic precision on that basis alone. But knowing full well that there's applications like this out there that are really cool and are going to help humanity, but are not necessarily, it's not necessarily going to work for me in developing a funding program if I do that. So that's it.
Great, thank you, Dina.
Yeah.
Okay, I'm running